This is the last session of the conference. I do appreciate everybody staying till the end. We have two final talks this afternoon. Hope you all had a good lunch break. The first talk is by Curtis Callan, who is the James McDonnell Distinguished University Professor of Physics at Princeton University. And he'll be talking to us about basically some topic involving the intersection of physics and biology, which is really something that many theoretical physicists often get interested in, going all the way back to Erwin Schrödinger, of course. So I'm very curious to see what goes on here.
Yes, it used to be that when a superannuated physics professor went off into something else, he went off into philosophy. Now they go off into biology. Okay, well, thank you for, as was said, staying around to the bitter end. I'm in the unenviable position of being not quite between you and the cocktail hour or escape, but also of following an absolutely bravura talk. You know, there's just no way that I can entertain you at the level that you were entertained this morning. Okay, so I was asked by Lars, asked is perhaps the wrong word, I was forced by Lars to give a talk at this conference, despite the fact that I have no history with Feynman other than that I spent the useful part of my career as a particle theorist, and I worked in fields that touched on Feynman's interests, and I certainly revered Feynman as a great scientist, and I learned from him indirectly many things. But you know, there was, in those days when I was sort of getting my way into theoretical physics, there was a kind of divide. Nowadays, there's a divide between urban and rural America. You know, the urban America votes Democratic, and the rural America votes Trump. In the 60s, physics was in a rather strange state. Nobody knew what the hell was going on, and there were religions. There was an East Coast religion, and there was a West Coast religion. And in fact, one of Feynman's colleagues at Caltech, I was told, referred to Princeton as the mysterious East. I was a Princeton denizen. I lived in the mysterious East, and I practiced bizarre meditational exercises like learning how to renormalize quantum field theory and things like that. And as I mentioned, my only actual direct interaction with Feynman, I think, was at one point during day one of the conference. It was a collision where I bounced off Feynman at some lecture at my undergraduate college. And maybe it was that made me decide I had two options. I could go to graduate school at Princeton, or I could go to graduate school at Caltech. And I had a really nice letter from the president of Caltech inviting me to come be a graduate student, but they had heard good things about me, and they would offer me generous financial terms. And I can't remember why, but I turned them down to go to Princeton. But it would have been great either way. But I missed the opportunity to bathe in the aura of Feynman and learn from him what so many of you have told me you learned from spending time either directly in Feynman's force field or receiving quanta from it indirectly and afar. All right, so Lars gave me two options. One was to talk about Feynman and biology. The other was to talk about Feynman and quote unquote society. Somehow, you know, everybody knows about the Challenger story, but Feynman thought deeply about many things. And as we heard from the letter that Michelle quoted from this morning, that he wrote from the Challenger Commission, he had a rather sophisticated view of what went on in the world and how science and scientists interacted with it. So I had a choice between trying to learn something about Feynman and society or Feynman and biology. I had no idea that there was much there in the subject of Feynman and biology, but it is a subject that I have taught myself something about, and I've been working in it as a theoretical physicist trying to do something useful in modern biology. for the last decade. So I figured I better take that path. So here's what comes out. So let's see if this works. I'm no historian, but I know that historians are supposed to go back to sources, and there are very few sources for Feynman and biology. So here they are. Let's see if I can do this. There's one actual paper, which I'll talk about in some detail. And then there are 3 other places where I found evidence that Feynman had thought about biology and worked on it. One of them is in the Feynman lectures on physics. I'll come to that. There is something that has been mentioned before several times. Feynman gave a series of lectures to the engineering staff of Hughes Aircraft, the company that put many, many satellites into orbit for various purposes. And he basically, I'm not sure how this lecture series came to be, but over the period of 4 or 5 years, I think he gave a series of really spectacularly wonderful lectures on all of science in some sense. And so I picked out the part that is on biology, and I'll tell you a few words about it. Then, of course, surely you're joking, Mr. Feynman has words about what happened, about his early higher educational career, and biology plays a certain role in that. And then finally, the document that's been mentioned more than once here, there's plenty of room at the bottom, that actually is inspired by and has ramifications for how biology should be done and how it might be done, in the future from the perspective of whatever the year was. It was close to 1960. Okay, I should also mention that I actually learned a lot from a paper that is published in some moderately obscure journal. A group of Italian physicists took the trouble to dig into the historical record and put together a compilation of their understanding of what Feynman was doing in biology when he actually devoted some serious attention to it. So, I wanted to make sure that this was noted. Okay, Feynman's adventures in biology, a timeline. So, am I being heard in the back row? Am I swallowing consonants and ends of words? I'm being reasonably audible? Okay. So, it begins, possibly before this, but evidence exists that in graduate school at Princeton, curiosity about biology led him to take a few courses, and he asked, Michel mentioned his course on, I'm not exactly sure what the course was on, but at any rate, I think it must have been neurobiology as then understood, and this is where the faculty member in question told him, yes, you can sit in on my course, but you must do the work. And I guess surely you must be joking, he mentions a homework assignment which involved the physiology of the cat and his going to the biology library and asking for a map of the cat so that he could know what all the bits and pieces, both, I suppose, bones, muscles, nerves, and whatnot, of the cat would be. And somehow at the end of all of this, he says, well, geez, I was able to figure out all this stuff, which is just memorization anyway, by going to the library in a couple of weeks. And these other people had to study the whole subject for years in undergraduate school and graduate school. What's the big deal here? So, however, that must have left him with an interest, or rather, the itch was not fully scratched. He also mentions, or it is mentioned in this book, his curiosity about other subjects, including philosophy and mathematics, and he also states how he taught himself, or how he interacted with mathematics. Namely, he would ask, well, what theorems are you proving? And they would tell him, as was said by, I think, Danny, well, imagine a generic blah, blah, blah with a this and that measure on it, et cetera, and he would say, oh, he means a circle. Then we can show that so-and-so. This means that two circles can have at most have either 0, 2, or infinite number of intersection points. Okay. and then from that follows blah, 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 which means that something that, if you thought about circles, couldn't be. So he says he was proud of himself because he could interact in a rather substantive way with the mathematics graduate students by letting them tell him about their deep stuff and translating everything down to something you could envision in your head and see when it was going wrong. In fact, I think he gave them a challenge. State me a theorem and I'll tell you whether it's true or false. And I don't know if this would work in mathematics now. Anyway, it was a long interval. 10 years later, he's a new professor at Caltech. And Caltech in the 50s, and probably from a little bit earlier than there, was in fact one of the meccas, one of the singular points of the development of modern biology. And this was due to the work of Pauling and Max Delbruck. And we'll talk about Max Delbruck in a bit. I guess starting when he arrived, which must have been roughly 52, Feynman was a regular in the lab meetings of Delbruck's group. And actually, if you follow the breadcrumbs, you'll find that the people who were youngsters in Delbruck's group in the early 50s, who eventually grew up to become famous molecular biologists who were asked to write retrospective articles and, autobiographical descriptions of what was interesting in their life. Feynman figures in many of those memoirs. He was such a scintillating presence, and so quick on the uptake and so enthusiastic that he impressed all of the people in the biology laboratories with his intellect. In fact, Jim Watson spent an unhappy year at Caltech the year after the DNA structure paper. And he in his memoirs, he said, and I met this remarkable person, Richard Feynman, and I tried to get him to work together with me on his next problem, which was discovering or working out the structure of RNA as opposed to DNA. And I think that didn't work out, but in any case, his interest in biology was quite intense at the time, and he didn't directly do any work in the lab, but he followed the developments of the burgeoning field of molecular biology. Then in '59, there's the Plenty of Room at the Bottom talk, and the headline is, it challenges physics to go to work at molecular scales imitating life. It foresaw the miniaturization revolution in computing, and it espoused a physics-inspired view of the core issues of biology. We'll come back to that later. Then in '61, '62, he took a sabbatical in place, working at the bench in Delbruck's lab. He studied reverting mutants of bacteriophages, and he was an author on a serious genetics paper, and we'll talk about that at greater length. And then that sort of was a summer project that eventually was absorbed into a large paper that got published in genetics. But what was really interesting to him was something which I can't quite figure out exactly what it was. It's hidden away in it's not clearly expressed in this genetics paper, but he got an invitation to come to Harvard to explain what it was that he had worked on or done, and followed up with further discussions with Watson, who was at Harvard at the time. And then in that year, he did further work with Messelson and Lamson on ribosomes, and these pieces of work targeted core issues of the new biology, but somehow they didn't quite work. And Feynman comments at some point that, I wasn't a professional biologist. I wasn't really taking this seriously, and I screwed up this experimental method, and I contaminated this reagent stock, et cetera, et cetera, et cetera. In '62 to '64, there's the Feynman lectures on physics. These dates may or may not be exactly right. The Feynman lectures had one biological chapter on color vision, And I will bring that up because in an aside, he showed that the insect eye optimizes its function as a device for seeing detail in the outside world by optimizing that function subject to the constraints, rather simple constraints of physics. And this foreshadowed a major future trend in biophysics, so it's worth mentioning. And then in '69, there were these Hughes aerospace lectures, and the year where he talked about biology and chemistry, well, there was a year, and I guess it was '69, where he talked about biology and chemistry, and they were pedagogical, but they were actually quite a masterful summary of what people knew at the time, and they contain Feynman's evolved view of the core problems of biology and some thoughts about how physics and biology should interact in the future. So I'll cover all of these in somewhat more detail. Okay, we all, how shall I put it, we teach 14-year-olds the basics of the molecular biology of life as a complete package. One understands there's DNA, it has a certain structure, it encodes it carries within it a code which is translated into protein, and the message, the code is how shall I put it, transported by RNA, which is copied from the DNA, and there's a machine called the ribosome, which makes the message into a protein, which then has function. It goes out into the cell, and it does the work of the cell. In the 1950s, even though the structure of DNA had been proposed, and I think determined is a proper word, by Crick and Watson in 1953, there was an enormous amount of uncertainty. People had no idea, well, no idea is the wrong word. There was no clear understanding of how the DNA was replicated, how the message inside the DNA was transcribed. There was a lot of, there are many things that we take for granted these days, which, and in fact, for example, I presume everybody in the audience knows that the genetic code is a triplet code. That is to say, in the DNA, there's a sequence of bases, A, C, T, and G, and it's every 3 bases that gets read as a particular amino acid. And it's a degenerate code, but but it's a good code, as somebody said. Okay, and all of these things took quite a while to get sorted out. The basics of molecular biology were very, very fluid at the time, despite the fact that the structure of DNA as the place where the code for life, in the sense of the code for proteins, must be stored. That was pretty quickly adopted and understood, but the details of how it all functioned were very, very much unknown. And it took a good decade to really sort that out. So much was known, but much was not known. So it was not, in a way, the best time for a physicist to get interested in biology, because there was a lot of really biochemical stuff that needed to get sorted out. Okay, but Feynman was interested, and he hung out in the lab of, I don't know exactly what that means. Did he go every day? I doubt it. Did he go once a week? Yeah, he probably went once a week to the group meetings and asked questions and, stirred the pot in the way he would stir the pot wherever he was. So Delbruck was the leader of the phage group, so-called. Well, not everybody here will know what a phage is, so I'll say a few words about that and try to explain why these things were interesting to people who were trying to figure out how the machinery of life works. So a phage is a virus that infects bacteria. Now, the viruses we normally think about are viruses that infect our cells of higher organisms, and they do various damaging things. There's another class of virus that infects bacteria, and they're not good for the bacteria. And in fact, back at the beginning, well, maybe around the time of the First World War, a French biologist had the good idea that phages, since they attack bacteria, would be good things to use to cure bacterial diseases. And this is a great idea whose, how shall I put it, elaboration was sort of short-circuited by the invention of penicillin, or the discovery, I should say, of penicillin and other such things. It's coming slowly back into focus. Anyway, why was this of some use? Well, the thing that they understood clearly was that the bacteriophage, the bacterial virus, was mostly DNA and not much else. Well, there had to be some proteins in order to make a package in which the DNA could be shuttled about. And the idea was that the genetic component, or the genetic composition of the bacteriophage, would be about as simple as you could get. And in fact, bacteriophages, our genome is 3 billion base pairs long. The length of the genome of a garden variety bacteriophage might be 100 kilobases or something like that. It's a lot, for the time, it would be very hard to manage 1,000, the information in 1,000 base pairs, but it's a lot more manageable than 3 billion. Okay, so what the phage group, what Delbruck's group was doing was essentially making a physical map of where genetic elements were located. So it turns out that the DNA of a phage it's either a loop or more commonly just a linear thing. And if a mutation happens at a particular place, some characteristic of the phage will change. And they had ways of looking at changes in the behavior of the phages. We see mutations in us by seeing different color eyes or this, that, or the other thing. In the case of the phage, you basically discover that if you make a mutation here, when you let it infect bacteria, it's more or less effective in infecting the bacteria. So by looking at what happens when you let the phage and the bacterium interact with one another, and sort of looking at the physical characteristics of the little region where a phage has infected a bacterium, and they would look at spots, basically, little plaques, they called them. Here's a lawn of bacteria, and somebody puts down a phage virus, and it infects one of the bacterial cells. The bacterial cell copies the DNA of the phage and makes 10,000 copies of it, and then bursts. And then there are 10,000 phages which are right there ready to infect the bacteria that are nearby. So you see little spots where something has happened, where a phage has had some impact. And by looking at different shapes, different consistencies and whatnot, they could identify different kinds of mutants. And then the really cool thing, which is where I guess maybe this is what got Feynman excited, was that, okay, so you find a phage that has mutated because the little plaque that it makes looks different from the way it normally looks. And then you take another guy, another mutant that you've discovered that makes what is apparently a different such mutation because the plaque looks different. Then the magic, which they glommed onto somehow, is that if you so you get you have these two phages that correspond to two different mutations in a gene. They didn't really know physically and geometrically what the gene was at the time, but if you let both of them simultaneously infect a bacterium, okay, so you have two different phages that have two different mutations in them, presumably at different locations on their DNA, they're infecting the same bacterium. Well, what goes on inside an infected bacterium is that the machinery of the cell busily replicates the DNA of the phage. It's a Trojan, well, it's worse than a Trojan horse, right? And so there's a sea of copies of the DNA of these two mutated phages floating about inside the bacterium. And the remarkable thing, which I try to indicate here, is that if you have two strands of DNA, and there's a little dot on this one at one place, and there's a little dot on the other one at some other place where something has been changed. If these guys are floating around next to each other, they're mostly the same except for these two little dots. And it happens frequently that if you have two stretches of DNA sitting next to each other that have the same sequence, they can cross over. That is to say, you can make an exchange. And this is what I'm trying to indicate here. So here's one genetic element that's normal. It's been mutated here. And here's another genetic element that's normal, and it's been mutated here. And the mutation in H is on the blue chromosome, and the mutation in R is on the red chromosome. And if they cross over, then I'm going to get, as an output, somebody where H, I'll put these two pieces together, and I'll put these two pieces together. So you could do a mixing of these things. Now, the frequency with which you will get this combined output, since it's got a different set of mutations, the plaques that it makes will look different. And you can identify that. And I spare you some details here. By essentially doing a census of how often this combination of the two mutations occurs when the DNAs are mixed together in this way, you get some measure of how far apart they are in physical space. Because, if they're very far apart, there are more places where the DNA can basically come together, match up, and decide I'm going to cross over. If they're very close to one another, then that doesn't happen so frequently. So what the poor graduate students and postdocs would do would be to create hundreds and hundreds and hundreds of little petri dishes in which there was a lawn of bacteria, and you would inoculate them with phage, and then you would watch what happened, and you would ask, in how many of these plates or in how many plaques did I see something new that corresponded to this, the crossover. Okay, so, and they did some remarkable stuff. I mean, let's see if I can remember the name. But anyway, this was the general idea of the work that they were doing, and they, over time, created a very complex, a very elaborate map of where different mutations were on this linear genetic element, which everybody presumed was DNA. Now, Feynman he decided to do a summer project. In some place I read that he said, well, every summer I got into a car and we drove somewhere different in the States. This summer I decided I'd stay at home and I'd drive in my mind to a different field of science, all right? So he then worked for the summer in Delbruck's lab, and it sounds, if you read it, like scut work. There was a new strain of phage that they were interested in, and he pitched in, and he did apparently the scut work. He had a particular goal, and the goal was something like this. Sometimes, one of these mutants, you'd isolate a mutant, and you'd let it propagate for a while without trying to cross it with anybody, and you might discover that it would suddenly disappear. The mutant would go away. So what's going on? So Feynman's goal was basically to find situations in which a mutation got, in effect, undone by a second mutation, and try to find instances where this happened, the rescue mutation was as close as possible to the original mutation. And they could figure out how far apart things were by this technology I was just talking about. So Feynman found 3 mutants, and here is a figure from the paper that was written in Genetics 47. This is Edgar, Feynman, Klein, Lolosis, and Steinberg. Steinberg is a famous molecular biologist. Edgar, I think, is perhaps less famous, but he was, a really serious molecular biologist. And Feynman found S1, S2, and S3. And he found out stuff about these mutations that definitely did not make it into the paper. Their goal in this paper was to help other experimental phage biologists by giving them a detailed map. I have 37 different strains that I can send you in a bottle if you want, and I know how far apart on the chromosome these mutations are. He had studied the so-called reversion of these mutants, and he found results. These results are not in the paper. It just says, here are these mutations. Now, 2 years later, it was realized that these revertants were a piece of evidence for the 3-base genetic code. And in fact, well, I'll try briefly to say what was going on. The idea was that people eventually figured out, well, it was beginning to become, how shall I put it, in the mindset of the molecular biologists at the time, that mutations could occur in a number of ways. A particular base could just get changed because some chemical action took place. A particular base could be deleted. It could be just removed from the string, or a more or less random base could be inserted. And so it turns out what Feynman was looking at was a phenomenon in which, here is a string of DNA. Something gets inserted right here. Where's the insertion? No, I'm sorry. Somebody gets inserted right here. So instead of ABC, ABC, it's ABX, CAB, CAB, blah, 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 blah. Now, when an insertion takes place, you basically screw up the reading of all of the triplets from that point forward. And it's pretty obvious that, well, if you've screwed everything, this is really a bad mutation that really kills the. You may ask, well, how did they study this thing if this really killed it? It turns out it didn't really kill it totally. It killed it as a useful phage on a particular strain of E. coli. There was another strain of E. coli which was much less sensitive to these mutations, so they could mix and match and follow what was going on. And you can easily see that, well, if you make a deletion somewhere near, in other words, I've inserted here and I've got everything out of frame, as they say, to the right of this point. Well, all it takes to put the rest of the genome back in frame is just to chop out a base here, okay? And so if you have something where you've added something undesirable and then later on you chopped out something further to the right, there will be a stretch of genome in the middle that's wrong, not doing the right thing, but it won't be forever. It'll just be a finite number of bases or codons that are messed up, and maybe the gene will function okay under those circumstances. So he was looking for, and he didn't realize it, he was looking for reversion of mutants back to more or less normal by this kind of mechanism. And he studied how close on the DNA the insertion was to the rescue deletion, and by crossing different mutants, he was able to say that they were very close, but in the absence of the triplet code, it was hard to be definitive. And I guess because they couldn't say something definitive, this information was not put in the paper. So in reality, he came pretty close to discovering something important, but it's not written down. There was enough buzz about what he had found, and he had sufficient cred amongst the biologists because of his previous history with interacting with the Delbruck lab, that Watson invited him to Cambridge to explain what he'd found, and he said that it was well received, and he was very happy, and he talked a lot with Watson, and they did a little experiment that didn't do anything, didn't go anywhere, and he came back home pretty pleased. But unfortunately, there's just no record of, okay, he came close to discovering something important, but it's just not written down anywhere, and I could not quite, how shall I put it, reconstruct what it was from the written record. For the rest of his sabbatical year, he focused on the key issues of the ribosome and protein synthesis. He isolated ribosomes from bacteria and came close to pulling off a classic experiment with another biologist, a woman who was actually quite distinguished in her later career. He was going to use bacterial ribosomes and plant messenger RNA to see that the bacterium would create the proteins that were appropriate to a plant. Okay, so this would have been a really classic experiment. But he says something about, well, I had lots of ribosomes in the freezer, but I'd messed it up somehow, and they were contaminated and no good. That's all we know. So he did some very interesting messing around and clearly was very intrigued by the whole thing. But, in this series of activities, he was not exploiting his core competency, which is really doing things that are in some sense mathematical in nature. He was doing what the biologists were doing, which is great. They were doing something important, and he found it just interesting and fun. Okay, so that's the one paper that he wrote or was involved in writing, and he has a very, very amusing story about. Yeah, he was told his colleagues wanted him to write things up, and he had some difficulty writing what he had found down in a way that the biologists could understand. And I think it just petered out, which is too bad. Okay, so let me pass on to the small is beautiful, or lots of room at the bottom story. We heard a lot about this already. Included in that speech is Feynman's view of the problems posed by biology. He says, the most central and fundamental problems of biology today, what is the sequence of bases in the DNA? What happens in a mutation? How is the base order in the DNA connected to the order of amino acids in the protein? What's the structure of RNA? How's it related? And so on. And that was indeed a correct statement of the problems at that time. It's very easy, says he, to answer many of these fundamental biological questions. You just look at the thing with a good enough microscope. And then he says, this is something that physicists could do, and if they wanted to be helpful, they would do it. And so this was a parallel track to, I could write information at the atomic level. And he says, well, look, biology also does that, right? They do write information at the atomic level, or at least the molecular level. And why can't we do that in an intentional way? Okay, so I'll come back. There's a bit of a manifesto in there, and I will come back to how his manifesto changed over time. So the next thing is biology in the Feynman lectures. So in the middle of the discussion of optics and electromagnetic waves, there are two chapters on how the eye works and how we perceive color. And it's interesting stuff. I think it's all correct. And a side note, I have heard from people, or at least one person who was a TA in the course, that, first of all, they could not understand why the students were having any difficulty grasping Feynman's inspiring presentations of classical mechanics or ordinary electromagnetism until they came to these lectures, where they hadn't any background. They didn't know anything about this. They were getting it from the first, these were topics outside their expertise, and for once, they were blown away by the Feynman fire hose, just like the students. So they appreciated what the problem is. Tucked away in the seeing chapter is a little gem, a demonstration that the parameters of the insect eye were such as to give the best possible performance subject to the constraints of physics, showing that evolution is a pretty damn good engineer. And the future would show that this approach illuminates many aspects of biology. Okay, so I think it's fun to look at this and see the equations are pretty simple. So the insect eye differs from ours in the sense that it's not one eye, but a whole lot of them. And so I don't know if we can see it here, but okay, so this is, I guess it's a housefly, and you can't quite see it at this level of resolution, but there are lots and lots and lots of little dots on this eyeball. Each dot is a lens, and the lens focuses light down a narrow tube to a neural element that turns light into nerve impulses. Okay, so basically each cone-like cell is looking just geometrically in some angle. It's collecting light that's coming in from a certain range of angles, okay? And that range of angles is just, well, what is the diameter of the lens at the top of the cone, and what's the radius of the eyeball, assuming that they all have their little, nerve endings down here. Well, it's d over r. That's the delta phi. Now, so why, you look at the eye and there's a certain size of those little cone cell, of those little amatidia, as they're called. Why not make them smaller and have more of them so that you can see more detail? Well, I don't know. Maybe the fly doesn't need to see detail. It just needs to see a blob that's our nose or something like that. Well, not so fast. You can't just make the size of the lens arbitrarily small, because light has a finite wavelength, and diffraction will fuzz the angular resolution of this individual cone by delta phi is equal to, roughly speaking, lambda over d. That is to say, the smaller the size of the lens, the bigger the diffraction effect that fuzzes things out. And so if you say, well, the true angular resolution of the ommatidium is roughly the sum of the geometrical resolution plus the diffraction resolution, and you say, okay, suppose I choose d so that this number is as small as it can be. There's a best size for the lens, and it's square root of lambda times r. And, cute, nice little argument. Again, it had been done before, but it was not widely known, I believe. So he actually says, and if you look at the numbers for the housefly, the answer is, it's really pretty nearly right on. 10 years ago, a neurobiologist by the name of Marlowe had gone to some museum collection of insects and done all these, he had the same idea. And he did the comparison. He got square root of head size, lens size, so that's square root of r this way, and that's d this way, and you plot everybody on, plot 20 different species on this plot, and you get for biology, a pretty damn good straight line, which is what this argument says. So that's rather cute. The numbers come out pretty close for many species. So evolution must know about physical law. Well, of course it does, but it's kind of interesting that it knows about physical law to the point of going to the absolute limit of, well, there's a function, right, which is seeing detail, and they get the best that they can. Okay. The Hughes lectures. This was a magisterial overview of the subject. I don't know, people in this room, if they're physicists, they probably hate organic chemistry, or they read about organic chemistry and they just can't remember this stuff. And I am afflicted by this, and I feel very guilty about it. I mean, I probably it's one of these things that one should have learned in one's youth, but I was, I don't know, repelled by it. So he actually goes through the basics of organic chemistry and how organic chemistry plays into or turns into metabolism and the Krebs cycle and the this and the that. It's just beautifully done. And I'm sure it would have been a joy to hear him saying it. And I'm absolutely certain that I would have been in an equal state of ignorance as I am now, an hour after having listened to the lecture. But you can always go back to the book and learn what's the difference between an alkane and an alkyne, an alkene, and so on. Okay, so, but I was interested in this from the point of view of, well, what does he think about the big question? What is biology and what is its relation to the other sciences? So, he says, to find an appropriate starting point is an impossible job. The subject matter differs from chemistry and physics in some significant ways. All the related fields of biology, to a great extent, are independent. You got to take a course, that's the problem. You've got to take n different courses to really get a grasp of the whole problem, the whole field of life. The reason for this is biology lacks a basic foundation of fundamental laws. Laws developed by theory and proven to be true by experiment. We'll find very little mathematics as we go through the material. Basic algebra and calculus will suffice. Lacking this underlying unifying thread, as in physics, I have found it most difficult to organize the material in a meaningful way. So he does the best he can. I've tried to orient the subjects on a scale ranging from the very small to the very large, where many complex biological systems interact in a closed environment. This is the field of ecology. So he explicitly says, we want to know what is the basis of life at the molecular level, and we want to work our way up to the not just the system level of an individual cell or an organism, but at the level of ecology. Thus, I plan sooner or later to span the biological spectrum from the submolecular level to the grandiose ecological level. Unfortunately, partons got in the way. If you look at the notes as they're available on the web, it really trails off into incoherence when you get beyond a sort of halfway there, which is really a pity, I must say. And so there was somebody who was carefully taking notes and reconstructing them, and I think the fact that Feynman basically gave up, he just said, I don't have time to do this, I'm working on partons, was sad. Okay, so that is roughly well, let me go one slide further into talking about Feynman's attitude toward biology. One of the things you can take away from this is how his view of biology evolved over the decade between Room at the Bottom and the Hughes lectures. So Room at the Bottom, as I say, is very much inspired by molecular biology, which was a work in progress at the time. And he takes inspiration from the nanomachines of the cell. He issues a challenge to physics, create the tools to see these machines, explains that there's no obstacle in principle to doing so, and he focuses on the molecules that do the cell's work and proposes to understand them essentially by watching them do their thing. Okay, there's a totally molecular biology in spirit. The Hughes lectures display a more systems view of what biology is about. Since there are no fundamental laws, he proposes to organize, of biology that is, he proposes to organize by scale. Curiously, he rather explicitly, he definitely does not argue that mathematics might help or be necessary, but he does say that life has to follow from the laws of physics. The lectures are amazingly comprehensive, compact, and up to where they stop, clear presentation of virtually all that was known at the time. There was even some immunology in there. So he worked hard on this. In preparation for each of these lectures, he went and read textbooks, read papers, and kind of organized it all in his head. And God, it would have been wonderful to have such a teacher. Okay, by '69, the time of the Hughes lectures, the foundations of molecular biology were pretty much laid, and a more predictive, quantitative, theoretical view of biology could have been imagined. The Hughes lectures give some evidence that Feynman was beginning to think along such lines, but he was hijacked by physics before he could develop such thoughts. Now, how might Feynman's thinking have evolved from there? Well, theoretical physicists have continued to be fascinated by biology, but the goals have evolved since Feynman left the subject years ago. Biology quickly answered the basic molecular questions and began to, quote unquote, look at how the molecular parts worked together to make a working cell. Leaving aside advances in microscopy, understanding cellular function posed a raft of basic questions that only physical science could answer. Diffusion and how bacteria sense their environment, how proteins find their correct folded state, why, even though the binding energy involved in copying DNA is alarmingly small, the error rate in copying DNA in a thermodynamic process, apparently, is extraordinarily low, much lower than a simple Boltzmann argument would mean. So, and after that, in addition to that, observations of collective cell movement, like in embryonic development, posed questions that could be addressed by the new physics of what's now called soft condensed matter. And beyond questions of physics and biology, advances in biology called for a systems approach to understanding how living systems could work. As Feynman actually says it in his Room at the Bottom lecture, biology is at bottom about information transmission, processing. It's about evolution. It's about doing things that are predictable in spite of noise. And this is not physics in the strict sense, but you can't pursue such matters without building mathematical frameworks. And this quest has become the heart of modern biophysics. And I'll try to explain this last point as best I can. I will now bifurcate, or rather, I will change direction and talk not about Feynman, but about what I think has been going on since Feynman left his breadcrumbs for us to follow. Now, I learned something from Danny Hillis's talk the other day, of which I was totally unaware, namely that he and Feynman actually realized that there was an interesting mathematical problem in evolution, in the, so to say, the theory of evolution, and they busily and very, I'm sure, quite brilliantly reconstructed the theory of neutral evolution that had been proposed by Kimura and Ota roughly 20 years before, I think. So, and I liked Danny's description of Feynman's response to that, namely, when told that what they were saying was true but not new, Feynman said, "Oh, great! You mean we're total amateurs and we actually got the right answer." That kind of summarizes the man for me. Okay, so the way I'm going to go from here to the end, wherever that will be, is going to be guided by a Poor Richard's Almanac. I don't know, in this audience, people don't know about Poor Richard's Almanac. Benjamin Franklin was a great statesman, but he was also a scientist of remarkable achievement. And he had, early in his life, he published something called Poor Richard's Almanac, which was a collection of wise sayings. So I'm going to pick several wise sayings that have biological import, or at least import for physicists who want to think about biology. The first is, it's very easy to answer many of these fundamental biological questions. You just look at the thing, okay? You know why you fellows are making so little progress? You should use more mathematics like we do. Now, he didn't really mean that. And he says, he then qualifies that with a statement, actually, there's no part of science where more rapid progress is being made than in biology, since, I don't know when he stated this, but it was sometime in the 50s. Biology is not simply writing information, it's doing something about it. There's nothing that living things do that cannot be understood from the point of view that they are made of atoms according to the laws of physics. What I cannot create, I do not understand. This wonderful little aphorism sitting up in the left-hand corner of his blackboard. And I think we saw several photographs of that blackboard, and there it was until it was finally erased. Okay, so these thoughts of a theoretical physicist inspired by biology remain relevant today, and they're convenient pegs on which to hang a discussion of what Feynman's lesser followers are finding today as they try to make sense of biology. Okay, you just look at the thing. I don't think I have to beat this one to death, right? It's been mentioned. The visualization challenge was met, and then some, by combining the tools of lasers, sophisticated optics, genetic engineering, computers. We can now look inside the cell and even go beyond the diffraction limit. So there's advanced microscopy. You can genetically engineer organisms so that, specific proteins that are expressed in specific organs can be seen by their fluorescence. You can engineer neurons to express calcium-sensitive dye proteins, and you can use this to watch individual neurons firing in a live mouse or worm. You can watch individual genes being transcribed into message. And then, of course, there's the whole sequencing revolution, the, so to say, observation or gathering of the at least raw information content of cells and other related things can now be done with remarkable efficiency. In short, we can now observe the inner workings of the cell in great detail. You should use more mathematics. Well, that I agree with, okay. So the flip side of our ability to look inside the cell is that the data we can now collect is becoming too copious to directly convey meaning. It's a little bit like what Maria was telling us about the detectors at the LHC. I mean, they have an even bigger problem, although, with some extrapolation, one can see that the biology problem could get almost as bad, and maybe the solution is going to be the one that Maria obviously loves a lot, but I'll have a word or two to say about that. Okay, the need for mathematical frameworks to make sense of what we see when we do our looking is really quite urgent. And so just some numbers. Current DNA and RNA sequencing, it's not just that you can get the genome of a species. You can get the level of expressions of thousands of genes in an individual cell for different growth conditions or points in the cell cycle. You can record from thousands of neurons in a behaving animal. You can see each neuron spiking. And you can do that for, neurons can spike every 1/10 of a second, and you can often record from, say, the brain or the retina for hours. And so you can do the math and figure out, how many bits we're talking about here. Single cell sequencing and expression level tracking for currently tens of thousands of cells. The idea that every cell in your liver is the same as every other cell in your liver, or every other cell in this particular muscle is the same as every other. It's just not true. And they are in fact, they may have the same basic genome, but they're doing different things. And so how the hell, and that different things that they're doing has some degree of variance, and somehow they all know, and the degree of variance can be quite substantial, and yet they know that they're liver cells or muscle cells, and they do the right thing. Some interesting things can be interesting questions can be posed by getting this degree of data. And so on, and so on, and so on. I was going to say single cell sequencing, synthetic biology. You can make millions of variations on a genome and try them out, see which ones do better at something. Okay, experimenters these days in biology can routinely collect giga, if not terabytes of data, and computers make it possible to store and at some level analyze such mountains of data. So, some pretty pictures which show some of the things that I'm talking about. It's worth mentioning the ever-growing suite of omes, genomes, exomes, proteomes, interactomes. They will sequence the DNA content of the bacteria in your stomach to find out how many different species there are, or on a patch of skin, and in principle learn useful things from them. It's even entering medicine. Cancer centers are now routinely sequencing in detail tumors that are taken from cancer patients. This has not yet done any good that I can see for the cancer patients, but it is on a track, one hopes, if there is understanding available at all, to actually having an impact on medicine. Okay, modern physics has a big data problem too. Maria has laid out what the problem is in particle physics. Let me just mention what we have when we're dealing with cosmology, the CMB. We have a map of the sky. We have a map of the temperature on the sky, the temperature of the microwave background. So the dark and the light spots are actually recording what is the variation in temperature on the scale of 10 to the minus 5 degrees Kelvin on the background 2.7 degree on average temperature of the universe. So there are millions of data points here, and they are processed by our cosmology friends into temperature fluctuations, how shall I put it, the angular content. They're Fourier transformed in angular space, and you see a plot like this. That is to say, the fluctuations on scale of 2 degrees are one thing, the fluctuations measured on scales of 5 degrees or something else, and so on and so on. And there's a red line, and the red line is theory. That is to say, we're able to take this collection of data and run it through our understanding of classical physics, general relativity, and so on, and come out with this. That is to say, oh, the universe at this time, at the time that these fluctuations were released to free stream in the universe, the energy content was so much dark matter, so much neutrinos, so much photons, so much atoms, and so on. We got some physically relevant information out of a rather random-looking set of points by having an underlying framework on which to hang it, okay? It wasn't just a question of using a statistics package to figure out what's going on here. Okay, we rely on a well-founded set of underlying mathematical laws. Unfortunately, for big biological data, the deep laws don't help very much, and we kind of have to satisfy ourselves with intermediate level principles. And it's not clear what they are. So let me just try to convey very briefly what I hope are moderately illuminating concrete examples of what I mean by these intermediate level principles. Okay, so modern biophysics is looking for theoretical approaches to comprehending the data that we've been talking about that have some kind of generality across the tree of life, even if we can't directly tie them to physical law. So the basic idea is to apply the theoretical physics method of understanding complex phenomena through the inference of simpler hidden and mathematically expressed mechanisms. I think I will go through one of these things. I don't have time to go further. So I was going to talk possibly about two threads, and naturally I oversubscribed. Thread one was correlated probability distributions on high-dimensional data and the problem of learning them from sparse data. These problems are everywhere. So a given visual scene produces a stream of spikes on the neurons that go from your retina to your brain, and that's all the brain gets. It gets a million, there are a million fibers. Each one of them is going beep, 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 sort of maybe 10 times a second. And I look at Frank, and he's looking directly at me, and I get one set of spikes. He turns down to look at something else, and I look at him, and I still see Frank, despite the fact that the stream of spikes on the neurons are totally different. Somehow, Frank is a probability distribution on this stream of spikes, and I'm somehow able to say that this particular set of stream of spikes, which I'm only going to see once, is drawn from the Frank probability distribution. How the hell you learn this stuff is really, 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 really mysterious. So that is a mine to be dug into. And I would say one of the threads of modern physicists' approach to biology is figuring, going at this in different ways, trying to get some generality of understanding. The other one that I was going to talk about, but will not, is information transmission problems. That's fascinating too. Even bacteria have to think, right? They have to decide on the basis of some kind of signals coming in from the environment that it's time to turn into a spore, for example. Life is not going to be good, and you'd better sort of curl up into a little ball and wait for things to get better. Well, they get some random battering of molecules on sensing organs on the surface, and somehow from that random battering, they extract information. So there's information coming in from the outside. It's used to change behavior, and there are just vast numbers of problems of interest there. Let's see. So I've hit the 1 hour mark. Do you give me another 10 minutes? Okay, all right. I will. All right, so let me talk about the probability distribution story briefly. This has to do with the immune system. Now, one of the dogmas of modern biology, which requires a statement of exception, is that every cell in your body has the same genome. It's the set of rules for making some 20,000 different proteins as might be needed. Now, your immune system, that is to say the set of cells in your blood, whose job it is to recognize and kill pathogens, violate rather dramatically that dogma. So the different immune cells, let's say the different T cells or different B cells in your blood, in first approximation are all different. What does that mean? Well, they have one really important gene, and that is a gene for a protein that folds up and goes to be expressed on the surface of the cell, and it's out there, and its job is to recognize this looks like a piece of a particular pathogenic bacteria, this looks like a piece of a viral protein coat, and I don't like it. So how does this happen? I mean, it starts with the same genome for sure, but somehow the mature T cells and B cells are in some sense all different, at least in this one gene. And the idea basically is the individual cells in your immune system are good for each one is good for some specific task. It can recognize one specific, or a cloud of a limited number of invaders. And our strategy apparently is to create many, many different soldiers, each one of which is good for one thing, and hope that we have covered the waterfront. So, how many of these soldiers do you need, and how is the diversity created? Well, the diversity is created in an interesting way. Namely, in the genome to start with, instead of having a full gene for this surface receptor, what you have is a bunch of modules, okay? It's like the Chinese restaurant problem. You pick one from column A, one from column B, and one from column C. So there are 20 options for one piece of the gene, 10 options for another piece of the gene, and 30 options for another piece of the gene. And apparently, some enzyme makes random choices of column A, column B, and column C, brings them together, chops out the intervening DNA, and makes a recombined gene, which obviously has some degree of combinatorial diversity at this point. That would not be enough. So what actually happens is that in addition, as I kind of indicate here, when these things are brought together and the intervening DNA is chopped out, two other things happen. There's a little bit of chewing away at the ends of these modules that are brought together. There's a little bit of random insertion between them. When you start putting all of that together, you have the potential, at any rate, for many, many, many different outcomes. Is it 10 to the 6 different outcomes? Is it 10 to the 10? Is it 10 to the 18? Just how bad can it get? All right, so the problem that some people, including me, set themselves was basically, can you, from the data that you can get, so the data that you can get is, we can take blood, or somebody with the appropriate license can take blood from a patient, separate out the immune cells, and in effect, using PCR, amplify the sequence of this gene that is responsible for the surface receptor, that's responsible for the specificity of each cell. And if you do this for 1 million cells, you'll get, when you do the sequencing, you'll get 1 million different sequences. So you can actually get the data. What is the sequence? What are the sequences that are in the immune system? And then the job we set ourselves was to figure out, from that, can you infer what are the stochastic rules for this process of you pick one from column A, you pick one from column B. Well, do you pick with equal probability all of the options, or is it somehow, are there some biases? You chew away. How many do you chew away? 0, 1, 10? Different probabilities for each. You insert stuff. How many? Different probabilities. So the question was, could you, by making use of some mathematical thinking, could you in fact infer from the data, which it's biggish data, not gigantic data, could you infer the detailed rules for generating this diversity? And then once you have that, well, you'll see what that's good for. Okay, so the answer is you can do it because you have a model for how this stochastic process happens. The model is, well, all I have to tell you is the probabilities for 3 or 4 different independent types of stochastic event. And so the basic idea is, well, think about, I'm choosing dishes from the 3 different columns, and suppose it's much more likely for me to choose the spring roll than the fried eel or something like that. What is in fact my the probabilities that the organism adopts? And so you have a model for what is the probability for generating a particular sequence, or what is the probability of a particular sequence of events that can produce a particular sequence that you observe. And then you play some statistical games to figure out what is the choice of these probabilities that maximizes the likelihood that you would see the set of sequences that you actually saw. All right, so that's too detailed for this. And you can actually do this. It works very nicely. And you discover that everybody has basically the same set of rules. But from the rules, you can learn that everybody has the same set of rules, but virtually nobody has the same cells, okay? That is to say, the diversity is so great that you can, we know how many of these immune cells there are in our body, and we can, from the probabilities of generating individual ones, we can figure out what is the likelihood that you and I will have one common T cell or 10 or whatever. And there are various other things that one can do with this. But anyway, the idea, and I just say one last thing, what we discover, we can, with all of this information, we can inform ourselves about what is the entropy of this distribution. If all of the things that you can observe were created with equal probability, how many of them would there be? And the answer is 10 to the 14 for us. For mice, it's a lot lower, even though superficially their rules look pretty much like ours, but they have a much less diverse immune system. Seems to be good enough for them. We don't really know why that is. But this is an example of one of the things you can try to do. Biology is not just writing information. Okay, I think I'm not going to go there. So that was there was an informational story, which I think is quite interesting, but it's a little bit further out on the edge. We've actually sold this story about understanding the rules for making individual T cells and what you can do with it. We've sold this to real immunologists. They kind of buy it. There's an information story that we've worked on which we have not successfully sold to real embryologists, but anyway. Okay, so I'm getting close, I'm almost at the end, just maybe one more slide. Okay, nothing that living things do cannot be understood from the laws of physics. So life is certainly governed by the laws of physics, but how to get from them to intermediate mathematical principles that we can use in practice is unclear. And what I just described to you and the other things that I might have brought up are warm-up exercises. And the relation to fundamental law is really tenuous. You just have to make use of we're making use of our attitude toward what constitutes science. There's a menu of less ad hoc questions we don't know how to attack yet, but that might be conceptually closer to the laws of physics. And it would be, one can hope that we'll make some progress on those sometime in the future. I just mentioned one. What is it about non-equilibrium statistical mechanics that makes it possible for populations of distinct, stably reproducing entities to arise? We have no idea, okay? But we have an existence principle. So developing this higher level of theory is developing the lower level of theory I sort of described as what theoretical physicists can do, and doing it is a worthy challenge for those who would like to emulate Feynman. And I have one last thought. This wonderful dictum, what I cannot create, I do not understand, which basically every theoretical physicist hopes that he can realize, namely, I don't have to go and look at the paper. Well, typically I have to go and read it at least once, but then later on I could go and sit down and recreate the essence of the argument and the result. Now, I fear that Feynman's appealing dictum is going to lose relevance. The physicist's ideal of explaining things by pencil and paper reasoning from fundamental law applied to simple abstract models, it already doesn't work in a lot of places in physics anyhow. And the problem is there are emergent phenomena that are not baked into the fundamental equations, but arise from strong correlations between the degrees of freedom we put in by hand. Confinement is a good example. We're not going to, maybe someday somebody will prove in some mathematical sense confinement, but the best we can imagine at the moment is that it's an output of lattice gauge theory simulations, and no single human mind is going to reproduce that. And this is one story among many. In the end, it's a question of what you mean by understanding in the scientific sense. I don't think the situation for biology is intrinsically any different than it is for, say, nuclear physics or condensed matter physics. And even mathematicians are beginning to accept computer proofs of complicated theorems. So the hell with philosophical problems. Let's just forge ahead and do what we can. And when we run out of gas, we'll become philosophers. But let's keep at it for a bit. Okay, so I'll just leave this slide up. I think there's an important future for people trained in theoretical physics, in thinking about biology. I think there's a vast field where they can do important things. Eventually, if they can, if it turns out that what we can do for biology is important, the biologists will teach their students what's necessary for them to do it straight away. We have a temporary competitive advantage, and I would suggest that we should exploit it. Okay, thank you.